Good evening, everyone. Today is the lecture 4 for the unit Cell, the unit of life. So, till now, in the earlier lectures, what we have studied is we get to know what is cell, what are the different organelles which are present in a cell, what is the difference between a prokaryotic cell and a eukaryotic cell, how a prokaryotic cell, what are the organelles which are not present in prokaryotic cells. Then we delve more into the eukaryotic cells, where we understand the differences between the plant cells and the animal cells. What are the major differences between these two? Then further we studied what is endomembrane system, what are the components of endomembrane system, and we studied the function of those components in detail. So today we will be studying the remaining organelles which are present in the cell. So starting with the mitochondria. So mitochondria was discovered by Kolliker in 1880. He discovered mitochondria in the muscle cells of the insects. Altman then named them as bioplast. Benda in 1897 finally coined the term mitochondria. Mitochondria is a plural term. For singular, we use mitochondrion. So, mitochondrion is a spherical or rod shaped cell organelle, as we can see in this figure, and it has two membranes. So, if you guys remembered it, in the first lecture, I gave a mnemonics to remember the organelles which have two membranes. So, it was namak, where N stands for nucleus, M stands for mitochondria, and K stands for chloroplast. So, one of the organelles which has two membranes is mitochondria. So, it has two membranes. The one is the outer membrane, and the other one is the inner membrane. The outer membrane, as we can see in this figure, is smooth, while the inner membrane has finger-like projections or finger-like enfoldings, which is known as cristae, as we can see in this figure. And the inner membrane has stalked particles, which are also known as Raker particles or F0, F1 particles, or we also call them as ATP synthase particles. So these particles help in the oxidative phosphorylation, and mitochondria helps in the aerobic respiration inside the cell. So therefore, the mitochondrial cavity is filled with the homogeneous granular mitochondrial matrix. Since the mitochondria is the site where the ATP synthase complex is present, and it helps in the generation of the energy inside the cell. It is also known as the powerhouse of the cell, or it is also called as the storage batteries of the cell, since it helps in the generation of the energy in the form of ATP. One of the important features of mitochondria is that it has its own DNA. It has its own genetic material in the form of a circular mitochondrial DNA, RNA, and 70S ribosomes. Plus, some of the proteins, enzymes, and lipids are present within the mitochondria. The mitochondria, just like the bacterial cells, divide by the fission, by the process of fission. The function, as I told, mitochondria synthesize and store the energy-rich molecules ATP, that is adenosine triphosphate, during the aerobic respiration. So therefore, they are also called as the powerhouse of the cell. So this was about the organelle mitochondria. The next is the plastids. Plastids were first observed by Schimper in 1885, and they are found in plants and euglenoids. Euglenoids are the organisms. Euglenoids are the organisms which can perform photosynthesis during the daytime and can use their flagella to procure their food, just like an animal cell during the night. So plastids are present in both plant cells and euglenoids. And plastids are classified based on the different types of pigments which are present in them. So they are classified into three broad classes, which is chromoplast, leucoplast, and chloroplast. So chromo, as I already mentioned in the first class, chromo is related to colors. So chromoplast, these are different colored plastids that contain the carotenoids. These are present in fruits, flowers, and leaves. And leucoplast are colorless plastids, and the main function of these plastids is to store the food materials. So examples of these plastids are amyloplast, amylo related to starch. So amyloplasts are the ones which store the starch, aleuronoplast that stores the protein, and ileoplast that stores the lipids. So these are the three basic types of leucoplast. The next is the chloroplast. We all know what is chloroplast since we study it from our basic, like from earlier classes also. So these are green colored plastids that contains the chlorophyll and helps in the process of photosynthesis. So these are the green colored plastids containing chlorophylls and carotenoids, especially carotenes and xanthophylls. As I mentioned also earlier, like chloroplast is also a double membranous cell organelle, and the matrix is called the stroma. As we can see in this figure, the matrix is called as the stroma, and the stroma has many membranous sac-like structures, which is called as thylakoid. We can imagine thylakoid as a single coin. So it has many sac-like structures, which is called thylakoids. And if I arrange one coin over the other and make a pile, so it will give us a appearance of what we called as a granum. So when we arrange thylakoids in a pile-like manner, in a like a pile of a coin, it arranged to form the granum. The grana are interconnected by the stroma lamella, as we can see. So these different grana are connected through stroma lamella, which are also known as intergranal membranes or stromal thylakoids. These membranous structures have photosynthetic pigments, for example, chlorophylls, carotenes, and xanthophylls. And they have major complexes like photosystem 1, photosystem 2, cytochrome B6F complex, and ATP synthase that helps in the process of photosynthesis. The stroma, just like, for example, just like mitochondria, the chloroplast also has its DNA. So it has a circular chloroplast DNA, RNA, 70S ribosomes, enzymes, and coenzymes that helps in carrying out the various activities within the chloroplast. And as I've mentioned, the main function of the chloroplast is that it helps in the process of photosynthesis. The next organelle is ribosome. So ribosomes are the non-membrane bound structures, and they are the granular structures which were first observed under the electron microscope as dense particles by George Pallade in 1953. So ribosome are composed of ribonucleic acid and proteins and are not bound by any membrane. Usually, the ribosome consists of two units. One is a larger unit, as we can see in this figure. So it is made up of two units. One is a larger subunit, the other is a smaller subunit. And the major difference, ribosome is an organelle that is present in both prokaryotes as well as in eukaryotes. But the major difference between the prokaryotic and the eukaryotic ribosome is that is between the subunits that assemble to form the ribosome as a whole. So in prokaryotic ribosome, the subunits are the 50S and the 30S. These 50S and 30S subunit assemble to give us the 70S ribosome in case of prokaryotes, while in case of eukaryotes, the subunits are little different from the prokaryotes. So in case of eukaryotes, the subunits are 60S and 40S. So these 60S and 40S subunits combine to assemble to form a bigger ribosome as a whole structure in the form of 80S ribosome. So this is the major difference between a prokaryotic ribosome and a eukaryotic ribosome. And it is very important to remember that the prokaryotic ribosome is a 70S ribosome that is made up of two subunits, 50S and 30S. And while eukaryotic ribosome is made up of 80S ribosome, is an 80S ribosome that is made up of 60S and 40S subunits. Here, S actually stands for the Swedberg unit, or we can say it is the sedimentation coefficient, and it is indirectly a measure of its density and size. So we cannot directly sum up and say that 50S plus 30S should give us 80S. No, it is not like that. So 50S and 30S will give us a ribosome, which is a 70S ribosome. Here, S is the Swedberg unit, and it is also known as the sedimentation coefficient, as I mentioned, and it is basically related to the density and size of the particle. So the main function of the ribosome is that it helps in the protein synthesis. So these are the sites of the polypeptide or the protein synthesis. We know that in case of prokaryotes, there are many ribosomes that can attach to a single mRNA and can lead to the multiple polypeptide chain formation simultaneously. So they lead to the structure formation, which is known as the polysome. So we studied that in the first class. So the main function of the ribosome is that it helps in the protein synthesis. And also we studied that when ribosomes are present on the membrane of the endoplasmic reticulum, they may give it a rough appearance leading to the, so we call that structure as a rough endoplasmic reticulum. And if it is absent, then it is known as the smooth endoplasmic reticulum. So this was about the ribosomes. So the next is the cytoskeleton. Cytoskeleton, as the name suggests, cyto is related to cell. Skeleton is so cytoskeleton is something that is giving the support to the cell. So just like our skeletal system. So it is a complex network of interconnected microfilaments and microtubules of protein fibers which are present in the cytoplasm. The microfilaments are composed of actin and microtubules are composed of tubulins. So the main composition of microfilaments is that actin, while the main protein of which the microtubules are made up of is the tubulin subunit. And as the name suggests, and as I also mentioned, the main function of cytoskeleton is that it provides a mechanical support to the cell. Also, it helps in the cell motility, cell movement, it helps in the cell division, and helps in the maintenance of the cell shape. So what are the different types of cytoskeletal filaments which are present? So these can be actin microfilaments, these are intermediate filaments, and these are microtubules. So actin microfilaments are made up of actin subunits and made up of actin protein. Microtubules are made up of tubulin protein, and intermediate filaments are made up of intermediate filament protein. Actin filaments are dynamic, they help in the movement. They are directional, that is, they have a polarity, they have a plus end, they have a minus end. They helps in maintaining the structure of the cell and allows the movement of the cell. And while in case of intermediate filaments, they are relatively static, that is, they do not move. They provide the structural integrity and the high tensile strength to the cell. In case of microtubules, which are made up of tubulin protein, they are dynamic, that is, they also move, they are directional, they also have a polarity, they are non-compressible structures, and they help in the formation of mitotic spindle during the time of cell division. So this is about the cytoskeleton, and the main function, as I already mentioned, main function is to provide the mechanical support, cell motility, cell division, and maintenance of cell shape. The next component is cilia and flagella. Cilia, for the term, the singular of the cilia is cilium, and flagella singular is flagellum. So are hair-like outgrowths of the cell membrane, and they help in the movement of this cell. Cilia are small structures which work like oars and causing the movement of either the cell or the surrounding fluid. While flagella, in comparison to cilia, are comparatively longer and are responsible for the cell movement. So cilia basically work like when we suppose we row a boat and we use the oars. So therefore, it helps in like providing the thrust to allow the movement of the cell, while flagella is the one that acts as a motor for the movement of the cell. So flagella are comparatively longer. They are relatively less in number as compared to the cilia. And the prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of cilium or the flagellum show that they are covered with the plasma membrane, and their core is called the exoneme. They possess a number of microtubules running parallel to the long axis, as we will see further. So exoneme, so this is the basically the section of the cilia, and we can see that the exoneme usually has a 9 doublets of a radially arranged peripheral microtubules. So these blue colored structures, blue colored ring like structures, are basically the peripheral microtubules that are present in the form of doublets, that is two present together. So peripheral, there are 9 such peripheral tubules that are present in the form of doublets, and a pair of centrally located microtubule. So we can see that these are the 9. Starting from here, we can see that these are the 9 doublets of peripheral microtubules, and one is present at the center, one pair. So such an arrangement of microtubules is referred to as the 9 plus 2 array. The central tubules are connected by bridges and is also enclosed by central sheath, as we can see highlighted by red in this diagram. So the central microtubules are connected by a bridge-like structure, and they are covered or enclosed inside a central sheath, which is connected to one of the tubules of each peripheral doublets by a we can say a spike-like structure, which is known as a radial spoke. So this central sheath is connected to the peripheral tubules through a radial spoke. So since there are 9 such peripheral microtubule doublets, so there are 9 radial spokes which are present that helps in making the connection between the peripheral microtubules and the central sheath enclosing the central microtubules. The peripheral doublets are also interconnected like this. There is an interconnection between these peripheral microtubules as well through the linkers. So these peripheral tubules are interconnected through these structures which are known as the linkers, and also the interdoublet bridge is present that helps in the connection that connects the two doublets of the peripheral microtubules. So this was the structure of the cilia. Both the cilium and flagellum emerge from the centriole-like structure, which is called the basal bodies. So both the cilia and flagella emerge from the same structure, which is a centriole-like structure called the basal bodies. So this was the basic structure of the cilia. So we can, again, if I'll repeat in short, so it consists of it has a 9 plus 2 array arrangement in which there are 9 doublets of peripheral microtubules which are present, while one doublet is present at the center. Also, it is the central doublet is enclosed inside a central sheath, and that is connected through the peripheral microtubules through a radial spoke. So there are 9 such radial spokes that helps in the connection. Then these peripheral microtubules are interconnected through the linkers and also through the interdoublet bridge. So this was about the cilia and flagella. However, there are some differences between the cilia and flagella. As I mentioned, cilia are small in size. They are 5 to 10 microns, while flagella are long. They can be up to 150 microns. As I also mentioned that cilia are more in number. They may be up to 14,000 per cell, while flagella are few in number. They may range from 1 to 8. So bacteria may have, like a cell may have a very few flagella ranging from 1 to 8 number of flagellas. The next is cilia beat in coordinated manner while flagella beats independently, and cilia take part mainly in locomotion and attachment, while flagella is involved only in the locomotion. There is no role of flagella in the attachment. So the main function of cilia and flagella was to allow the movement of the cell and helps in the attachment of the cell with the surface. The next is the centrosome and centrioles. They were discovered by von Benden in 1880. Centrosome is found in animal cells, and in some motile algae, it is absent in plant cells. This we studied when we were studying the differences, when we were learning the differences between the plant cells and the animal cells. So there we understood that centrosomes are not present in the animals, are not present in the plant cells. They are absent in the plant cells. And centrosomes are present near the nucleus. It has two cylindrical structures that are called the centrioles that are surrounded by a less denser cytosol called the centrosphere. So this is the structure of centrosome, where we can see that centrosome has two cylindrical structures that we called as centrioles, which is shown in the right side of the figure. So these centrioles are usually lie perpendicular to each other, and these are surrounded by a less denser cytosol called the centrosphere. So this is about the centrosome, and the centrosome is made up of two cylindrical structures, which is called the centrioles. So centrioles are arranged at right angles to one another, as we can see in this figure also. So they are present perpendicularly to each other. Each centriole is made up of a wall of 9 triplets of microtubules. So just like we studied the like arrangement of microtubules in case of cilia and flagella, similarly, these microtubules are arranged in a pattern which is a wall of 9 triplets of microtubules or cartwheel structure, which we call in case of centrioles. So in case of centrioles, these are 9 microtubules and they have a triplet fibril, which is A, B, and C. And A of the triplet is connected to the C through a CA connective. Also, like these A, B, and C microtubules run perpendicular to each other. So when we look into when we look from upside of these centrioles, we'll find that these A, B, and C microtubules are running perpendicular to the triplets of microtubules are running perpendicular to the each other, forming a cartwheel-like structure. So the main function, the adjacent microtubules are connected by the proteinaceous strands, and they have 9 spokes in total, connected from the central rod, or which is also known as the hub. So there are 9 spokes, like 1, 2, 3, 4, 5, 6, 7, 8, and 9 spokes in total. So that are connected through the central rod. The main function of these of these centrioles is that they form esters and organize the formation of spindle fibers during cell division. So you might have studied cell division. So during cell division, there is spindle formation. So these spindle formation, like centrioles, play an important role during that. So they form the esters and organize the formation of spindle fibers during the cell division that further helps in the arrangement of chromosome, so that further the cell division can take place. Also, they are involved in the formation of cilia, flagella, and axial filament in the sperms. So these are the main functions of the centrioles. Again, centrioles are present only in animal cells and they are absent in the plant cells. So this is also an like important aspect or important point to be noted here. And the main function was that it helps in the spindle formation during the cell division. The next is the nucleus, which is also known as the karyon. The plural is nuclei. So we know that nucleus was discovered by Robert Brown in 1831, and it is a darkly stained material inside the cell. It is the largest cell organelle which is present in eukaryotic cells, especially in case of animal cells. As we know that in case of plant cells, it might be vacuoles that occupies most of the space inside the cell. So in case of eukaryotic cells, especially in case of animal cells, it is the largest cell organelle which is present, and it is usually spherical, while usually it is spherical, but shape may change in case of some cells. For example, it may be lobed in case of WBC or kidney shaped in case of paramecium. Usually single nucleus is present inside a cell, but in some cases there might be two nucleus there which is present inside the cell. Such cells are known as dikaryotic, since two nucleus are present. So karyo word is related to nucleus. So whenever it is said like dikaryotic, so di means two. So that organism has two nucleus. So usually the nucleus is spherical, but it may be lobe shaped or kidney shaped depending on the cells also. As I mentioned, nucleus is also the third organelle which I mentioned that has a double membrane structure. So it is a double layered nucleum. It is a double layered membrane structure, outer membrane, inner nuclear envelope, inner nuclear membrane, and there are pores which are present in between these, which are known as the nuclear pores. Also, a transparent granular matrix called the nucleoplasm or karyolim is filled inside the nucleus, and chromatin network composed of DNA, histones, and a darkly stainable spherical body called nucleolus. So, in short, what is present in the nucleus? So, nucleus is a double membrane structure. Outer one is known as the outer nuclear membrane, and inner one is known as the inner nuclear membrane. Then there are pores which are present in the nuclear membrane. So these pores are known as the nuclear pores. The fluid which is filled inside the nucleus is known as the nucleoplasm. And beside this, there is a structure which is called as the chromatin. So chromatin are nothing but they are the condensed form of the chromosomes which are present. So during different stages of cell divisions, there are different stages of chromosomes also. Like in some, like at some stage, there will be chromosomes will be highly condensed, while on the other stage, they will be loosely present inside the cell. So basically, chromatin is nothing but the condensed form of chromosome that consists of DNA and histone proteins. These histone proteins help in the binding of the chromosome, leading to the structure formation, which is known as the chromatin. And these chromatins, and all beside this, there is a darkly stainable spherical body which is present inside the cell, inside the nucleus itself, and it is called as the nucleolus. So the cells having nucleus are called nucleated or eunucleated cells. The cells which lose nucleus at maturity, for example, we know that mammalian RBC or sieve tubes in case of angiosperms, these cells lose nucleus when they mature. So these are called as enucleated cells. The cells having incipient nucleus in which the nucleus is basically diminished nucleus, or we can say that the very like primitive nucleus where as such nucleus is not present, DNA is present in the naked form. So such organisms are called prokaryotic cells, example bacteria and nostoc, and the cells having well defined nucleus are eukaryotic cells. We know that in case of higher plants and animal cells, the nucleus is well defined and they are a part of eukaryotic cell, and they come under the category of eukaryotic cells. So what are the functions of a nucleus? Nucleus is the controlling center of the cell. It is the main unit where the protein formation, protein synthesis may take place. So basically it contains the genetic material DNA, which regulates the various metabolic activities of the body by directing the synthesis of structural and functional proteins. So basically, when we will study further about the DNA proteins, we will get to know that DNA gets transcribed into the RNA, which gets translated into the protein. So it is the DNA which is present inside the cell that regulates various metabolic activities, directing the synthesis of structural and functional proteins. So since most of the proteins, the formation of proteins is dependent on the DNA, which is the genetic material, which is present inside the nucleus. Therefore, nucleus is the main controlling center of the cell, controlling all the activities of the cell, controlling the protein formation inside the cell. So this was about the nucleus as we studied chromatin. So to understand chromatin further, we should know what are chromosomes. So interphase nucleus, as I mentioned that during different phases of the cell cycle, there are chromosomes are present either loose, they may be present loosely or they may be present in the bound form. So interphase nucleus has a loose and distinct network of nucleoprotein fibers called as chromatin. These chromatin are highly stained, like darkly stained material, and the material of nucleus, these chromatins are stained by the basic dyes, for example, acetocarmine, and it was given the name chromatin by scientist Fleming. During different stages of cell division, cells show structured chromosome in place of the nucleus. Thus, chromosome is highly condensed form of chromatin. So we can say that from this figure, it is very clear that what I actually mean by chromosome, what I actually mean by chromatin. So this is the DNA, which is present inside the cell. This DNA is very long. For example, it may it may be up to 2 meter in length, but it is difficult to accommodate this 2 meter of DNA inside this small cell, which may range in microns maybe. So this DNA is then bounded through the histone proteins. These histone proteins, along with the DNA and some other non-histone proteins, form a compact structure which is chromatin, and these chromatin further form a more complex or more compact unit which is known as the chromosome. So we can see that and our DNA is therefore present in the form of chromosome inside our cell. So basically we see that the chromosome is highly condensed form of the chromatin, and chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also some RNA. So chromosomes can be best studied at metaphase stage because size of the chromosome is shortest during the metaphase. And if we want to study the shape of the chromosome, we have to study the shape at the anaphase stage. So at different stages, like if we have to study if I have to study the size of the chromosome, like if I want to count the number of chromosomes, or if I want to understand how the chromosome looks like, so the best stage at which I can study the chromosome is at the metaphase stage. The number of chromosomes which are present in a gamete is called the genome. So basically, we know that we are diploids. So there are two sets of chromosomes which are present inside our cell. So the number of chromosomes which are present in a single set is known as the genome. So for example, like we humans have 46 number of chromosomes. That is, we have 23 pairs chromosomes. So the total amount of genome, total amount of gene content, or the number of chromosomes in a gamete, that is, the gamete only contains the single set of the chromosome. That is, in gamete, n is equal to, like in gametes, the number of chromosomes which are present in a gamete is 23. So that will form a unit which is known as the genome. And a single human cell has approximately 2 meter long thread of DNA distributed among its 46, that is 23 pairs of the chromosomes. And every chromosome visible only in dividing cells essentially has a primary constriction, like they have a primary constriction at the center, or which is also known as the centromere, on the sides of which disc-shaped structures which are known as the kinetochores are present. So every chromosome has a constriction at the center, which is known as the centromere. This is the primary constriction, and this is known as the centromere. And from these centromere, from like on the sides, we can see there are disc-like structures which are present. These are known as the kinetochores. So from centromere, the kinetochores are present beside the centromere in a form of a disc-like structure. Centromere holds the two chromatids of a chromosome. So during cell division, when we know that there is a formation of sister chromatid, so this is the centromere that holds both the chromatids. So this is the chromatid 1, this is chromatid 2, which are held by the centromere in a chromosome. Also, based on the position of the centromere, like where this is present, the shorter arm, the centromere may divide the chromosome into two arms. The one arm may be short, the other arm may be long, or both may be equal as well. So the shorter arm is known as the P arm, from the term petite, which means small. So the shorter arm is known as the P arm, while the longer arm is known as the Q arm. So based on the position of the centromere, like where the centromere resides inside the, like where this where this primary constriction is present. So based on that, either both the arms may be equal in size, there may be a shorter arm and a longer arm. So based on the position of the centromere, the chromosomes can be classified into four types. They may be metacentric, submetacentric, acrocentric, or telocentric. So metacentric, as the name clearly suggests, like it is the centromere is present just in the middle of the chromosome, forming two equal arms of the chromosome. So, like there is no short arm, no long arm, both the arms of the chromosomes are equal in size. While in submetacentric chromosome, the centromere shifts a little upwards, creating a shorter arm and a longer arm. But the difference between the shorter arm and the longer arm is not much, as we see that in case of acrocentric, the in case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short arm and one extremely long arm. So this is the difference between submetacentric and acrocentric. That in case of submetacentric, uh, there is a formation of a short arm and a longer arm. But in case of acrocentric, since the centrosome is present more close to its end, therefore one arm is extremely short as compared to the other. And the last is the telocentric, where the centromere or the where the centromere is present at the just at the terminal. So there is a very short arm, which is like short arm is the may or may not be visible. So due to the presence of the centromere, which is at the terminal of the chromosome. So these are the four types of chromosomes based on the position of the centromere, that is metacentric, submetacentric, acrocentric, or telocentric. Sometimes a few chromosomes have non-staining. This is very important, that the secondary constrictions, the primary constriction was centromere. The secondary constrictions may be present in few chromosomes, and these are non-staining, that is, they do not take up the stain, and they are present at a constant location. And this gives the appearance of a small fragment called the satellite. So we can see in this figure. So this chromosome has a secondary constriction which is present at the like beside the primary constriction, that is the beside centromere. There are secondary constrictions which are present, and these are known as the satellite. These small fragments, since they appear like a satellite, so these are known as the satellite. The secondary constriction is non-staining and found at a constant location, as I mentioned, and it is also known as NOR. NOR stands for nuclear organizing region. So these like secondary constrictions are also known as NOR. And in humans, these are present in what number of chromosomes? In 13, 14, 15, 21, and 22. So in case of humans, these secondary constrictions are present in 5 chromosomes. Chromosome number 13, 14, 15, 21, and 22. So this was about the chromosomes. So the main function of chromosome is that it contains the genetic material DNA, and it helps in the transfer of genetic information from one cell to the from one generation to the next generation during the time of cell division. The next and the last organelles which is present in the cell, inside the cell, are the microbodies. These microbodies are very minute membrane-bound structures that may contain the enzymes for the metabolic activities. These microbodies can be of various types like peroxisome, spherosomes, glyoxysomes. So peroxysomes helps in the breakdown of long chain fatty acids. They are involved in the peroxide metabolism in the animal cells, and they are involved in photorespiration inside the plant cells. Spherosome helps in the synthesis and storage of lipids, while glyoxysomes, as the name suggests, they convert the fats into the carbohydrates. So these are the 3 main different types of the microbodies that are present in the plant cells and the animal cells. So peroxisome, that helps in the breakdown of long chain fatty acids and involved in peroxide metabolism in animals, while in case of plants, they are involved in photorespiration. Spherosome that helps in the synthesis and storage of lipids, and glyoxysomes that converts the fats into the carbohydrates. So this is all about the different organelles which are present inside the cell. I hope all of these things are clear to you, and if there is any doubt, I'll be happy to take up those doubts either today or in the next session. So basically what we studied today is, so we first studied what is mitochondria, then we get to know what are plastids, what are different types of plastids, what is the function of chloroplast, what are ribosomes, what are the different subunits of ribosomes which are present in a prokaryotic cell and eukaryotic cells, and then what is the cytoskeleton, what is the composition of components of cytoskeleton, what is cilia and flagella, what is the basic structure of cilia, and then we studied what are centrosomes and centrioles, and then we studied nucleus, which is the controlling center of the cell, and the chromosomes that are present inside the nucleus. So based on this, I have certain questions to discuss. So the question number 1 is, powerhouse of the cell is option 1 mitochondria, option 2 ribosome, option 3 Golgi complex, and option 4 endoplasmic reticulum. So we know that it is mitochondria that contains the ATP synthase complex that helps in the synthesis of ATP. Mitochondria is the site of aerobic respiration inside the cell that helps in the formation of ATP. And ATP is the basic energy, is the basic unit that provides the energy to the cell. So in a way, mitochondria is the powerhouse of that cell that helps in the generation of energy. Second is, infoldings of inner mitochondrial membrane are called. So option 1 is cristae, option 2 is cytoplasm, option 3 is crystal, option 4 is matrix. So we know that the outer membrane of the mitochondria is smooth, while inner mitochondrial membrane has finger-like infoldings, which are known as cristae. So the correct answer is option number 1, cristae. Question number 3, which of the following are plastids? Option number 1, chloroplast, option number 2, leucoplast, option number 3, chromoplast, option number 4, all of the above. So we know that chloroplast are green colored plastids, leucoplast are colorless plastids, and chromoplast are colored plastids that may be present in fruits and in leaves. So all of these are the kind of plastids only. So the correct answer is all of the above, since chloroplast, leucoplast, and chromoplast all are a type of plastids. The next question is, thylakoids are arranged in stack-like piles of coins called stroma, matrix, granum, or quantosome. So we know that thylakoids, which are coin-like structures, they are arranged in stack-like piles of coins to form the structure which is known as the granum. So the correct answer is the granum. While stroma is the matrix, which is the stroma is the matrix of the chloroplast itself. So the correct answer is that the thylakoids are arranged in stacks like the piles of coins called granum. Which organelle is not surrounded by any membrane? So we know that it is the ribosome which is not surrounded by any membrane, while mitochondria, lysosome, chloroplast are all surrounded by membranes. So the correct answer is option number 4, ribosome. The next question is, arrangement of microtubules in the core of cilium is. So we know that cilia consist of doublets of microtubules, peripheral microtubules, 9 doublets of such peripheral microtubules, and then there is a central pair of a microtubule which is present. So such an arrangement, we know that it is known as the 9+2 arrangement or 9+2 array arrangement. So the correct answer is option number 2, 9+2. Self-duplication or fission does not occur in. So option number 1 is mitochondria. We know that mitochondria do undergo fission. Option number 2 is centrioles. Yes, centrioles also do divide by the process of fission. Option number 3 is chloroplast. So as I told earlier in the class lecture, during the lecture also, that chloroplast also divide by the process of fission. And option number 4 is ribosome. So Actually, ribosome does not divide by the process of fission. So the correct answer to this question is option number 4, ribosome, as it does not self-duplicate or it does not divide by the process of the fission. Question number 8. Chromosome is highly condensed form of. So we know that first we go through the options. So option number 1 is chlorophyll. Option number 2 is chromatin. Option number 3 is rRNA. And option number 4 is microtubules. So we know that chromosomes are nothing but they are highly condensed form of chromatin. Chromatin along with chromatin that consists of histone proteins, non-histone proteins, RNA, DNA, all of these components leads to the formation of chromatin, and these chromatin further condensed to form the structural unit which is known as the chromosome. So the correct answer is chromosome is highly condensed form of a chromatin. Question number 9, which of the following is correct for secondary constrictions? So we know that in a chromosome, there are two types of constrictions. The one is primary constriction, the other is a secondary constriction. Secondary constriction may or may not be present, while the primary constriction, which is known as centromere, is present in the chromosome. And so let's see what the option says about the secondary constriction. So first is that they are non-staining. Second is they are found at a constant location. And third is they are NOR, that is nuclear organized regions. So option number 4 is all of the above. So we know that secondary constrictions are non-staining, they do not take up the stains, and also they are found at a constant location, at the constant location on the chromosomes, and they are a part of NOR also. So option number 4, that is they are non-staining, found at constant location, and NOR, that is they are nuclear organized regions. So all of these options are correct, and therefore option number 4 that says all of the above is correct answer. Question number 10, centromere is at middle in which chromosome? So we know that centromere, which is the primary constriction, which is present in the chromosome. Based on the position of the centromere, chromosomes are divided into 4 types as we study. So they may be metacentric, submetacentric, acrocentric, or telocentric. So it is the metacentric in which the centromere is present at the center, thus dividing the chromosome into 2 equal halves. While in case of submetacentric, there is a shorter arm, there is a longer arm. In case of acrocentric, the shorter arm is highly reduced. So the size of a shorter arm is very less as compared to the longer arm. And in case of telocentric, the centromere is present just at the the shorter arm is almost not visible. So therefore, the correct answer where the centromere is present at the middle. So the correct answer is metacentric. So with this, I would like to end today's session. I hope you would have get to know what is a cell, what are the different organelles which are present inside the cell, what is the function of each and every organelle, how is different organelles function in association with each other, just like we see in case of endomembrane system, like how these organelles help each other in packaging, manufacturing, packaging, and exporting the components in and outside the cell. So like what is the center of the cell, that controls the activity of the cell. So these are the different organelles, and this is all about the unit, the cell, the unit of life. So with this, I would like to end this chapter today. And if there are further any doubts, feel free to ask me like whenever it is convenient for you. Thank you.